Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
In a milestone for biomolecular design, a team of scientists has succeeded in creating new proteins that adopt one of the most complex folds known to molecular biology. These designer proteins were shown in the lab to spontaneously fold into their intended structures and embed into lipid membranes. Reported in the journal Science, this research opens the door to the construction of custom nanoscale tools for advanced filtration and DNA sequencing. Right now scientists all over the world are using protein nanopores as part of their effort to sequence genetic material from the pandemic coronavirus and discover mutant strains, said lead author Anastassia Vorobieva. For this project, we wanted to design new nanopore proteins completely from scratch that could serve as starting points for a wide range of future applications, including improved DNA sequencing. Vorobieva is a recent postdoctoral researcher in the laboratory of David Baker, director of the INST. Bacteria are encased in a specialized membrane, called the outer membrane, which protects them from the outside world. Proteins that embed into these membranes facilitate the movement of specific chemicals into and out of the cell. Such natural protein pores share a similar nanoscale structure, a flat sheet of protein that curls in on itself to form a barrel, through which other molecules, including nutrients, vitamins, and even strands of DNA, can pass. This is known as a transmembrane beta barrel. To create new transmembrane beta barrels, Vorobieva and colleagues used molecular design software to draft possible structures. Although they drew inspiration from proteins found throughout the living world, they arrived at sequences that differ from any known before. Their most successful designer proteins contain 8 ribbon-like strands that fold into a compact barrel structure that stands just 3 nanometers tall. We began with a relatively simple notion about what would make the proteins fold, said Vorobieva. But when we tested these initial hypotheses, nothing worked at all. That was very frustrating. We didn't assume we would get it right the first time, but we did think we could get some information back that would tell us how to move forward. Instead, I had to go back and look carefully at how nature solves this problem. The key was to try to detect patterns in those proteins. It was a really difficult thing to do. Researchers in the laboratory of Sheena Radford, Astbury Professor of Biophysics at the Astbury Centre for Structural Molecular Biology at the University of Leeds in England, tested whether improved versions of the designer proteins could embed into artifact. They found that they could do so efficiently without the help of any accessory proteins. This is in marked contrast to how natural transmembrane beta barrels fold. These designed proteins are interesting from a basic science perspective because they have no evolutionary history, said Radford, a specialist in protein folding. By studying them, we can discover some of the essential features that enable transmembrane beta barrel proteins to fold into a membrane. Banyang Liang, an assistant professor working within the laboratory of Lucas Tam at the University of Virginia School of Medicine, used nuclear magnetic resin. This work is the latest achievement in the rapidly progressing field of protein design. In recent years, scientists at the Institute for Protein Design have created innovative vaccines, experimental cancer treatments, and sensors capable of detecting antibodies against COVID-19. The ability to design new proteins from scratch with new functions has implications for diagnosing and treating a wide range of diseases, as well as for advancing materials science. With this type of research, it helps to understand a bit about how evolution works at the molecular level, but we are also trying to see beyond that. That's really the challenge of protein design, said lead author Vorobieva. The research team included scientists from UW-Medicine, University of Virginia School of Medicine, University of Leeds, Johns Hopkins University, and The Ohio State University. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.